Chapter 1, Human Biology, Science and Society, Screencast 2. This screencast is going to focus on an introduction to science and the process in which how science data collection is gathered or how we acquire knowledge. When we look at science, we want to think about science as knowledge about the natural world. One of the things that you'll notice quite often, science is broken up into different categories: natural sciences, social sciences. We're going to be focusing on natural science. In particular, we're going to be focusing on biology, which is the study of life, and in even more detail going into aspects related to human biology. Another thing that we'll be looking at into this screencast is how we actually acquire or gather that knowledge, and it's through a very logical step-by-step process known as the scientific method. Now, I'm sure many of you have studied the scientific method in many different classes. I was talking to a student yesterday. He's taking 3 different classes, and they are all presenting the scientific method at this point in the course. And one of the things I want you to be aware of is you're probably going to see different steps. Overall, the process is the same, but a number of individuals or different authors are going to name the steps differently. And I'll kind of address that as we go through this. For this particular class, we're going to use these 5 steps. The first step, observe and generalize. Step 2, formulate a hypothesis. 3, make a testable prediction. 4, experiment or observe. And 5, modify the hypothesis as necessary and repeat. As I said before, different authors are going to present this process in different ways. For example, this author chooses to present 2 and 3 as separate steps. In previous classes that I've taught, those 2 steps, 2 and 3, actually kind of meld together. So we'll be walking through these, and I do understand that if you have a different definition or something like that on an exam that's a short answer question, I definitely will work with you with regards to that, as long as the order is correct and it fits an actual logical step of what the scientific method is. But it's a process. It is a very logical process, and so I want to make sure that you are comfortable with that concept. The first is observe and generalize. And I want to make a point here because quite often one of the most common mistakes here is people jump the gun, and they quite often will list step 2, formulate a hypothesis, as the first step. And that's not the case. All right, you really can't formulate a hypothesis unless there's been some observations made. So that's really important. Okay? So the first step is to observe and generalize. Now, the textbook uses some terminology that I think is rather confusing. And you're going to notice that I've crossed it out here. Inductive reasoning. Later on, we'll see deductive reasoning. And there's a figure in our textbook that I find to be very, very confusing that doesn't add to the understanding of the scientific method. So I'm not going to go there. Okay. But here we're talking about an observation. So something that you see in the natural world has gained your interest. And then you make some type of a generalization. The examples that our texts are using first as an observation: every winter in the past was colder than the preceding summer. Okay? Notice that's specific to a single set of seasons, one winter, one summer. Next comes the generalization. Now, with the generalized statement, winter will always be colder than summer. So now they're generalizing that all winters, not just one winter-summer sequence, but all winters will always be colder than summer. So you should be able to understand that difference between an observation and a generalization. Now, the second step is formulate a hypothesis. Quite often, you'll hear a hypothesis described as an educated guess. And so what we're looking at here is a tentative statement about the natural world. Now, I'm also going to kind of look at the next step, which is make a testable prediction. Quite often in the past, other courses that I've taken, they've included some of these characteristics as part of hypothesis formulation. So one thing when we talk about hypothesis, it should be tested under multiple conditions, because once again, we're looking to see how general this particular statement may be. Also remember, the hypothesis is a statement, not a question. And so one of the things I want you to think about, the hypothesis is actually formulated in the form of an if-then statement. And many of you may have been trained in the past to write a hypothesis in that manner. A perfect example of a valid hypothesis would be, if I take this medication, then my blood cholesterol levels will decrease. So there's the if-then component. It's testable, and that's also really important. Okay, so when we look at these hypothesis/predictions, it needs to be specific and something that you have the ability to test. The next step is to experiment and observe. And as I noted previously, there's different types of sciences. We have the natural sciences, such as biology, chemistry, and so on. We have social sciences, like psychology and sociology. Regardless, they're going to use the scientific method. Now, how a prediction is tested is through either observation and experimentation. A number of my friends in graduate school were field biologists, so their research quite often was based on observation of the presence of a particular animal species or something along those lines. Okay? When we talk about chemistry or biology, we think of an experiment as a carefully planned and executed manipulation. We will be talking about the differences between controlled experiments and some examples of those. So typically when we talk about an experiment, we'll use the term experimental design, and that has some specific parameters that we'll address a little bit later. Okay? So when we use the term controlled experiments, all but one variable is accounted for. So you're looking to see if that particular variable is responsible for an outcome or what it is that you're testing for. The last step is based upon the data collected, modify the hypothesis as necessary, and go back to the previous steps. If the prediction is false, the hypothesis must be modified. If the prediction is true, only one part of the hypothesis has been tested. So further testing is required to support the entire hypothesis. And notice the term that I'm using, support. Hypotheses cannot be proven true. They are only supported or disproved. Now here, I want to walk through this particular diagram because I think it's really important, and it describes what I talked about previously as a experimental design. So first off, look what they're starting with, a large number of appropriate subjects. So this particular study is looking to investigate the impact of a drug on blood pressure. So you want to have a population where you might expect to see high blood pressure. You would not want a bunch of young, healthy individuals to make up your subject pool. So there's where that term appropriate comes in. The next step is where that original pool is subdivided into two groups. Here we look at group 1, and notice group 1 is given the treatment, group 2 is given a false treatment or a placebo. So that is the control group. They're going to be used as the standard of comparison because they have not received any treatment. So after a period of time, group 1 has taken the drug, group 2 has not taken the drug. Now, one thing I want to kind of get back to, these subjects don't know whether they're getting the drug or not. So that is known as single blind. Okay? Now, an even more stringent experimental design is what's known as double blind. in a double-blind experiment, not only does the subject not know what group they belong to, neither does the primary investigator. So groups have been determined from an outside party. So after a period of time, blood pressure is taken, and the purpose here is to determine whether or not the drug was effective in controlling blood pressure. So the question that you're going to ask at the end is blood pressure lower in the group that received the actual drug? If yes, then your hypothesis is receiving support that this drug actually did what it was supposed to. If the blood pressures are not lower or not different than the group that received the placebo or the control group, then that says something was wrong with the effectiveness of that particular drug. So the hypothesis needs to be modified to determine new findings. In this particular situation, there's going to have to go back to the drawing board and see what it is about the drug that did not work. This is an animation related to the scientific method. And as I noted previously, if you go to Blackboard on lecture materials, the PowerPoint presentations that are available will include these animations. I also have included a separate PowerPoint presentation that has just the animations for a particular chapter. So we won't be reviewing these animations as part of the screencast. Now, the next part is, how is the information from a particular experiment going to be disseminated, or how is the public going to know about the project or the research? In science, the gold standard is what's known as a peer-reviewed journal. Peer-reviewed journals, experts actually review and approve the articles and research project before publication. The other thing to keep in mind here is, when you look at these particular experiments, it's assumed that the results are specific to the conditions under which this experiment was actually done. Generally, peer-reviewed journals are the gold standard in how scientists present their findings. Now, another thing that's a part of the process is, how does a hypothesis become a theory? First off, it's a well-tested hypothesis. We're also going to look at something that's broad in terms of definition, has been extensively tested, supported over time, and actually explains a broad range of facts. And most importantly, a high degree of reliability. And what that means is, when other people do the same experiment, the hypothesis is actually supported also. Okay? Now, one thing that we have to keep in mind, and this is where technology plays a very important role, we are now capable of doing more things because of the tools we have available. So there is the possibility that some theories may be refuted because we now have the technology to go into or investigate that particular theory in greater detail. A really good example of a theory would be what started off with the hypothesis that all living things are made of cells. That has now developed into the cell theory, and it has withstood years, if not hundreds of years, and is true, regardless of what we know, regardless of the technology. That is an example of a theory that has not been refuted and has been true across a long period of time. Now, one of the hopes that you will get out of this particular class is the ability to understand scientific information in society. The expectation is not that you go to peer-reviewed journals. Typically, the information related in them is far more advanced. It's very specific to the science involved. But where we are in a course for non-majors, your primary source will be science magazines, nonfiction books. There's general interest news magazines, daily newspapers. For example, the New York Times every Tuesday does Science Tuesday. NPR has a show on Fridays, Science Friday. So those are good examples. But probably where many of us get our information now is the internet. And I have to caution you because you have to remember anyone can post anything on the internet. And you have to learn the skills of what on the internet is actually true and what is not true. Now, another goal of this course overall is that you develop the skills to be a critical thinker. And these are the primary things we want you to develop as you take this course. Become a skeptic. Don't believe everything you hear or everything you read, especially when we're talking about the internet. Learn how to read graphs. This is going to be especially important. Quite often in science textbooks, articles, there is going to be a graph that needs to be interpreted, a figure, and you remember that old saying, a picture is worth 1,000 words. So a lot of information can be held or found in a particular graph or figure. Understanding and appreciating the value of statistics. Math 160 MCC is Statistics 1, and it is actually a course that many programs at MCC actually want you to take. And statistics are really important. In the beginning, it may appear to be a different language because it really kind of is. And if you were to go to a peer-reviewed journal and look at the results section, you might really struggle reading and understanding that part of the study if you have not taken a statistics class. Another important skill that you should be developed is to distinguish anecdotes from scientific evidence. So for example, let's say that you have a friend and they've been taking the supplement, and this supplement is supposed to result in magical weight loss. Okay? If you just pay attention to your friend, that's really anecdotal evidence. There is no science to support the fact that that supplement actually does what it says it's going to do. So that's really important. Okay? The difference between facts versus conclusions. Conclusions are drawn. That's an important thing based on the data that you are presented. The other thing is the difference between correlation and causation. Just because you identify a relationship between two variables, it does not necessarily mean that one has caused the other. So that's the difference between correlation and causation. Now, in our world, science plays a very critical role. One of the things that we know, advances in science result in improved technology. The better technology that we have, it's going to improve overall the human condition. Now, science isn't the answer to everything, and it does have some limits, and a lot of those are based on the scientific method and the manner in which the information has been collected. Now, this last statement, and I've said before that I really don't want to be political, but we now live in a society where people are very skeptical of science. They are more willing to believe these various theories that are out there that are not based in science. And to me, that is a danger to our society and public health. One of the most important roles of science in society is to help us make informed choices. And I do want us to keep that in mind because I think that is especially important in the age that we live in now.